The day has been long. You feel it in your shoulders, in the specific weight that gathers behind your eyes by late afternoon, in the low hum of obligations that has been playing beneath everything since the moment you woke, and your mind, before your eyes were even fully open, had already begun cataloging what needed to be done. Meetings that stretched past their purpose. Decisions that required more of you than the people asking seemed to understand they were asking. On the particular fatigue of competence, of being the person who knows what to do, who does it, who has been doing it repeatedly and reliably for decades, and who still comes home at the end of the day to find that dinner needs making and someone needs something, and the work does not end because the location changed. Set all of that down for a moment, not as a command and not as a platitude offered by someone who does not understand the weight of it. Set it down because the story that begins now requires you somewhere else entirely, somewhere the obligations of this century cannot follow, somewhere on the southern coast of Africa. Approximately 150,000 years ago, the air smells of ocean salt and sun-warmed rock, and the faint mineral sharpness of low tide retreating from stone. The temperature along this coastline holds at around 18 degrees Celsius through most of the year, cool enough to be comfortable by tropical standards, warm enough that the glaciated interior stretching north and east is another world entirely. The sky above the sea cliffs carries no contrails, no satellites, no light pollution from any human settlement anywhere on the planet. Just the uninterrupted, unwitnessed blue of a sky that has existed for 4 billion years and will continue to exist long after every building in every city you have ever walked through returns, grain by grain, to the sediment it came from. A woman is moving along the shoreline below those cliffs. She is not young. Her face carries the specific marking of years lived in wind and light and salt air, the kind of marking that has nothing to do with vanity and everything to do with presence, with having been on a continuously and attentively in the world. Her hands know what they are doing without any instruction from conscious thought. She is not searching. She is going where she always goes, to what she already knows is there, carrying knowledge accumulated over decades that the younger women in her group have not yet had enough seasons to possess. She is doing this, specifically, instead of bearing another child, and that single biological fact, that she stopped reproducing and redirected everything she had toward the survival of what already existed, is in a direct and measurable sense the reason you exist today. Here is the counterintuitive fact at the center of everything this documentary investigates, stated plainly before anything else. Of the approximately 6,000 species of living mammals on Earth today, only 6 are known to experience true menopause, a complete, to genetically programmed cessation of reproductive capacity, occurring at a specific life stage, while the individual continues to live for decades afterward, remaining physically active and socially significant within their group. 6 species, out of 6,000. That is a frequency of 1/10 of 1%. And those 6 species are not biologically related to each other in any way that would explain a shared, inherited trait. They did not receive this biology from a common ancestor. The evolution arrived at the same radical solution. Stop reproducing, keep living, keep contributing, independently in at least 5 separate evolutionary events across approximately 90 million years of completely divergent biological history. The 6 species are human beings, killer whales, short-finned pilot whales, false killer whales, narwhals, and belugas. The last 5 are all toothed whales. The first is you. Scientists analyzing genetic data from approximately 200,000 women published results in 2021 in the journal Nature Genetics, identifying 290 specific genetic variants associated with the timing of menopause onset. 290 separate locations in the human genome, each contributing to the precise moment when the ovarian cycle concludes. The majority of those variants are concentrated in DNA repair pathways, specifically in the biological systems that govern how aggressively the body maintains the integrity of DNA in the cells, responsible for egg development. The timing of when your body stops producing eggs is written into your genome at hundreds of specific measurable coordinates. It is not random. It is not an accident of living longer than evolution anticipated. It is a program architecturally sophisticated, maintained by selection pressure sustained across thousands of generations. Programs of this genetic complexity do not persist in a population by oversight. They persist because they conferred a survival advantage large enough and consistent enough across enough generations and enough crises to outweigh the direct reproductive cost of building them. The question this documentary investigates, across the evidence of genetics, paleoclimatology, behavioral ecology, primate biology, and 40 years of killer whale observation, is why that program was written at all. What force was powerful enough, and what conditions were specific enough to make stopping reproduction in the middle of life more adaptive than continuing it? The answer reaches back approximately 150,000 years to environmental conditions, severe enough that the entire genetic future of every human being who would ever live, all 8 billion of them, was compressed into a small coastal population that may have numbered fewer than 10,000 contributing individuals, the size of a small town. The entire reproductive future of the species in one place on one stretch of African coastline, surviving conditions that had eliminated every other human population on the continent. What those individuals had that others did not, and specifically what the oldest women among them contributed that younger women biologically could not, is the answer to why menopause exists. The central mystery that this documentary will hold, develop, and finally resolve is this: menopause, by every standard definition of evolutionary fitness, should not exist. Natural selection filters with extraordinary consistency for traits that increase reproductive output. An individual who stops reproducing before death removes herself from the genetic competition entirely, and any genes that contribute to that early exit should, over sufficient generations, be progressively selected against until they disappear from the population. The theory behind this prediction is not speculative. It has the weight of 160 years of evolutionary biology behind it, tested against thousands of species across every class of animal life, and it holds. And yet menopause persisted. It deepened. It accumulated 290 genetic variants. The theory says it should not be there. The genome says it is. Resolving that contradiction requires understanding the specific historical conditions that made it adaptive and the specific role of older women in those conditions. Neither the biology nor the conditions are what conventional accounts of menopause suggest. This documentary moves through that evidence in a sequence that builds toward a complete answer. It begins with the world as it existed before the crisis, the climate, the landscape, the structure of small human groups along the southern African coast, the biological baseline of women living within those conditions. It builds through the genetics of bottleneck populations and what modern mitochondrial DNA reveals about who survived and why. It examines the biology of the menopausal transition itself, not as pathology, not as loss, not as the body's apology for having outlived its reproductive purpose, but as a program of precise and deliberate biological redirection toward a different function. It finds the same solution independently executed in the ocean, to where killer whale grandmothers have been studied for 40 consecutive years, and where the data is now specific enough to read as a biological argument written in behavior and mortality statistics. It arrives eventually at the complete answer, an answer specific enough to name the conditions that produced menopause, and profound enough to change how you understand every cell in the body you are living in right now. If you are settling in for sleep, let this be the final conversation of your day. The voice will stay steady throughout. The pace will remain unhurried. The information will reach you whether your eyes are open or fully closed. There is no urgency in what follows. What happened 150,000 years ago already happened, and the women who made the decisions that kept the human lineage alive already made them. What remains is only the understanding. Wherever you are in the world tonight, whatever coastline, or whatever city, whatever quiet room receives you, you are the specific, measurable consequence of something that borders on the extraordinary. The biology that carried your ancestors through the nearest thing to human extinction, that the fossil and genetic record preserves, is the same biology you carry right now, in this body, in this moment, in this life. This is its story. The woman foraging along the tidal pools at sunrise is about 34 years old, which means she has already lived through more concentrated loss than most people in the modern world encounter across a full lifetime. 2 children who did not survive their first winters, both gone before they developed the specific weight in her arms that would have meant they were going to be all right. For a season of droughts so severe that the tubers in the inland savanna shriveled to nothing in the ground before they could be harvested, forcing the entire group to move 40 kilometers north along the coast to find shellfish beds dense enough to sustain 26 people through a season that came within weeks of ending them. And she has watched the sky and the sea and the inland vegetation for 3 decades, with the specific quality of attention that develops in a person whose survival depends on what they notice. Not anxious attention, not vigilant in the sense of being frightened, but the calm, continuous, unconsciously processing attention of someone for whom the natural world is not a backdrop, but a text read daily for information that directly determines what tomorrow holds. She knows this stretch of coast, and she knows the caves where the prevailing wind comes from the wrong direction in winter, and cold air pools against the back wall, making them uninhabitable past a certain depth even when the entrance looks sheltered. She knows the tidal pools that refill fastest after a storm because of how the cliff face deflects the incoming water. She knows the specific rock formations where mussels concentrate in densities worth harvesting, and the ones that look similar but are not. And she knows these things not because someone told her once, but because she learned them over years, tested what she was told against what actually happened, corrected the errors, and survived long enough to correct them more than once. The southern coast of Africa during this period, approximately 150,000 years before the present, carried a specific combination of ecological resources that appears, from the accumulating archaeological record, to have been critical to human survival during a period of increasingly severe inland deterioration. The coastline near present-day Mossel Bay, in what is now the Western Cape province of South Africa, offered year-round shellfish, marine plants, occasional fish in nearshore pools, and seasonal migratory birds, all within a few kilometers of cave systems cut into the sandstone cliffs by millennia of wave action. So these caves provided natural protection from the wind and temperature extremes of glacial period weather. Archaeological excavations at the site now designated Pinnacle Point have recovered systematic evidence of shellfish consumption, beginning at least 164,000 years ago. The accumulations of shell material in the cave deposits, dominated by brown mussels, limpets, whelks, and some barnacles, are not random or opportunistic. They show consistent selection of specific species in specific size ranges, consistent return to the same collection sites across seasons, and a pattern of exploitation that implies not casual scavenging, but organized, knowledge-guided harvesting by people who knew exactly what they were looking for and exactly where it was most productively found. Shellfish, as a food source, carry a specific combination of properties that made them disproportionately important in the survival economy of small coastal groups. They are calorie-dense relative to the effort required to harvest them when the harvester knows the best locations. A woman with specific knowledge of a productive tidal flat, knowing the tide timing, the optimal harvesting depth, the species distribution across the flat's microhabitats, can gather enough protein and calories in 2 to 3 hours to sustain 2 additional people through the day. A woman without that specific locational knowledge, working the same stretch of coastline for the same duration, collects a fraction of that yield. The difference between these two outcomes is not the physical capacity to harvest. It is the knowledge of where to go. So, in conditions where the daily caloric margin separating adequate nutrition from deficit is measured in hundreds of calories per person, that knowledge difference is not a minor advantage. It is survival itself expressed as geography. Group size during this period, estimated from both the density of archaeological site remains and systematic ethnographic comparison with contemporary hunter-gatherer communities studied in analogous ecological conditions, so clustered around 25 to 50 individuals for a functioning residential band. The Hodza of north-central Tanzania, among the most extensively and rigorously studied living hunter-gatherer communities in the world, operate in groups of roughly this scale under broadly comparable ecological constraints. Within a group of 25 to 50 people, every individual contributes to the collective food economy in ways that are directly felt by every other member. The incapacitation or death of one highly knowledgeable woman does not represent a statistical fluctuation in group productivity. It represents the immediate, unambiguous loss of years or decades of accumulated practical knowledge that cannot be reconstructed from memory by anyone who did not spend those years accumulating it alongside her. Knowledge of this kind does not survive in abstracted form. It requires a living person to hold it, deploy it in real conditions, adapt it to new circumstances as conditions shift, and transmit it to the next generation through years of apprenticeship, in which the learner watches the expert act, and then acts themselves and is corrected, and then acts again. The world before the breaking was not a world of simple or peaceful existence. The archaeological record from this coastal zone offers evidence of sophisticated behavioral modernity that predates what was previously thought to mark the emergence of distinctly human cognition. At Blombos Cave on the South African coast, researchers have recovered evidence of an organized ochre processing workshop operating approximately 100,000 years ago. Multiple grinding stones, ochre pieces, marine shells used as containers, all arranged in ways suggesting a deliberate, systematic production process. At the same site, approximately 75,000 years ago, human beings produced cross-hatched geometric drawings using ochre crayons on silcrete stone flakes, the oldest known drawing by any member of the human lineage. These people were cognitively modern in every sense that matters. They thought abstractly. They communicated in language complex enough to coordinate group behavior across seasons. They maintained social identities requiring symbolic expression. The woman foraging at the tidal pools at 34 was not a pre-human figure moving through a simplified biological existence. She was a fully modern human being with a fully modern mind, living under conditions of resource stress and climatic uncertainty that demanded everything that mind could do. The biological baseline for women during this era requires some reconstruction from skeletal evidence, ethnographic analogy, and what is known about the physiological effects of the nutritional and activity conditions of hunter-gatherer life. Menarche likely occurred around 15 to 17 years of age, 2 to 4 years later than in contemporary well-nourished populations, because lower body fat percentages and higher energy expenditure from daily foraging activity delay the hormonal threshold that initiates the reproductive cycle. First pregnancy likely occurred in the late teenage years. Interbirth intervals of approximately 3 to 4 years, maintained by the ovulation-suppressing effect of extended breastfeeding under conditions where weaning foods were nutritionally incomplete, meant that a woman who survived to adulthood and remained in reasonable health could expect to bear somewhere between 5 and 8 children across her reproductive years. Of those children, perhaps 2 to 3 might survive to their own reproductive years under typical resource conditions, somewhat fewer during the severe stress episodes documented in the paleoclimate record. Maternal mortality from childbirth was not negligible. Skeletal evidence from this period consistently identifies young adult women as a high-stress demographic, and mortality risk associated with repeated pregnancy accumulates in the body in ways the bone record preserves. What the biological baseline reveals, when held against the ecological requirements of group survival, is something that deserves to be stated without qualification. The women who were most consequential to the group's survival in any given season were not the youngest. The youngest women were reproductively active, contributing to the next generation, but constrained in their other contributions by the demands of infants and young children who required continuous care. And the women with the most complete and tested knowledge of the group's specific ecological environment, the precise geography of food sources across seasons and years, the management of illness and injury, the social memory of intergroup relationships that could be called on during displacement, were the women in their 30s, 40s, and beyond. This is not a sentimental observation about the value of experience. It is a direct, quantitative inference from what knowledge requires in terms of time to acquire, and from the practical arithmetic of survival in a group small enough that every individual's knowledge contributed measurably to the collective daily caloric balance. Something worth holding as this chapter closes, because it shapes everything that follows. The question of why menopause produces significant biological disruption. The hormonal instability, the disrupted sleep, the systemic physical changes of the transition is a genuine evolutionary question. If menopause is adaptive, why did selection not produce a smoother, less costly version of the same outcome? Why does an adaptation that supposedly increases survival come packaged with years of genuine physiological challenge? The answer to this question is not a simple one, and it is not the one most commonly offered. It requires understanding what the biological disruption is a signal of, what system is being reorganized, and why that reorganization was so consequential to so many generations of women, that it persisted in this demanding form, rather than being smoothed away by selection over the same period that built the 290 genetic variants underlying it. that question opens in what follows, and the biology that answers it reaches directly into your own body. A finding in the population genetics literature deserves more attention than it typically receives outside specialist circles, and it provides the most direct available link between the biology of individual women alive today and the events of 150,000 years ago. Mitochondrial DNA passes exclusively through the maternal line. Every mitochondrion in every cell of your body, and there are hundreds to thousands of them in every cell, generating the chemical energy that powers every biological process you will undergo today, descended from your mother's mitochondria. Hers descended from her mother's, hers from her mother's before that, in an unbroken chain of maternal inheritance extending back through every generation of human beings who ever lived, to a single woman whose mitochondrial lineage is the direct ancestor of all mitochondrial lineages present in every living human being on the planet. This woman is referred to in the population genetics literature as Mitochondrial Eve. She lived in sub-Saharan Africa. Estimates of her date range from approximately 150,000 to 200,000 years ago, with the balance of recent analyses converging toward the lower end of that range. She was not the only woman alive at that time, not the first woman, not a supernatural ancestor, not even necessarily the most important woman in her own generation. She was the one whose maternal line never went extinct. Every other maternal line from her era eventually did, one by one, in the exact kind of catastrophic attrition that the paleoclimate record documents from this period. The structure of the human mitochondrial family tree reflects the history of those extinctions, the divergence of her lineage into macrohaplogroup L, specifically the split between the oldest branch, haplogroup L0, and all later branches. Laos L, 1 through L6, represents the deepest branch point in the human mitochondrial record. The San people of southern Africa, often referred to in the genetics literature as one of the most ancient living human populations, carry haplogroup L0 in its highest frequencies. When geneticists map the geographic distribution of mitochondrial haplogroups across every living human population on the planet, the oldest and most deeply branching lineages, the ones whose divergence from the common ancestor was earliest, cluster with remarkable consistency along the southern and eastern coasts of Africa. This is not the geography of where humans eventually spread. It is the geography of where humans survived when everywhere else failed. The map of ancient lineages and the map of coastal refugeia overlap with a precision that cannot be produced by chance. In 2021, a genome-wide association study of approximately 200,000 women of European ancestry, published in Nature Genetics, identified 290 specific genetic variants associated with the age at which menopause begins. This number requires some context to appreciate fully. 290 separate genetic locations, each contributing to the timing of a single biological transition. For comparison, the human height, one of the most extensively studied polygenic traits in all of human genetics, influenced by factors spanning nutrition, skeletal development, hormonal systems, and structural proteins across virtually every tissue in the body, is similarly polygenic in architecture. The genetic complexity underlying menopause timing is comparable to the genetic complexity underlying height. Traits with this degree of polygenic architecture do not develop casually or quickly. They accumulate over many generations of sustained selection pressure, with each contributing variant spreading through the population because it slightly increased survival or reproductive success in the individuals who carried it. 290 variants do not accumulate under genetic drift in a small population over short time spans. They accumulate under selection, sustained and directional, across thousands of generations. The variants themselves are distributed across the genome in ways that reveal the biological systems most directly under selection. A substantial proportion cluster in DNA repair pathways, specifically in the biological mechanisms that govern how efficiently the ovarian follicle cells repair accumulated genetic damage. Follicles that carry more DNA damage are eliminated by cellular quality control systems as unsafe for reproduction. Follicles that carry less damage survive longer. So the rate at which this attrition occurs, and therefore the age at which the follicular reserve is effectively exhausted, is partly determined by how aggressively the body's DNA repair systems operate in ovarian tissue. Women with particularly efficient DNA repair in ovarian cells experience later menopause. Women with less aggressive repair experience earlier onset. The timing of menopause is therefore, in part, a genetic program for the rate of ovarian resource management, a program that was adjusted by selection across the same period that produced the bottleneck signatures visible in the mitochondrial record. The implication is worth stating slowly. The timing of your own menopause, whether it comes earlier or later in your 50s, whether the transition is compressed or extended, is partly determined by genetic variants that were selected during exactly the period this documentary is examining. Not ancestrally, not in some general evolutionary sense. Specifically, the population that experienced the selection pressure that shaped those 290 variants was the same compressed coastal population whose mitochondrial descendants are every human being alive today. The women whose groups survived the mega-drought episodes passed their genetic variants forward. The women whose groups did not survive did not. The selection that accumulated those variants was operating on the same timeline, in the same geography, under the same environmental pressures, as the selection that shaped everything else about modern human biology. Your menopause timeline is a specific product of that specific period. She's a 2016 paper by Kristen Hawkes and colleagues in PNAS, titled Genomic Evidence for the Evolution of Human Post-Menopausal Longevity, examined whether the genetic signatures of grandmothering behavior, of a life history in which post-reproductive women provide significant help to grandchildren, could be detected in the modern human genome. The analysis found patterns consistent with selection for extended post-reproductive survival, each with the genetic signal strongest in populations, with the longest documented histories of the grandmother-intensive social structures that the grandmother hypothesis predicts should drive the evolution of post-reproductive longevity. The genetic record is not just telling us that menopause exists. It is telling us something about why the post-reproductive period is as long as it is, and what the selection pressures were that made several more decades of post-reproductive life worth the genetic investment to maintain. Consider what this means for the mitochondria in your cells right now. They are not passive participants in your biology. They are the direct cellular descendants, through an unbroken maternal chain, of mitochondria that were operating in the bodies of women who lived on those coastal cliffs and survived the worst that the climate of 150,000 years ago could produce. The women whose mitochondria these are did not survive because they were lucky or because they were stronger than the women whose lineages did not persist. They survived because they were in groups that had something the failing groups lacked: a specific configuration of knowledge, caloric contribution, social intelligence, and ecological expertise that was disproportionately concentrated in their oldest female members. The mitochondria you carry are the cellular inheritance of that configuration. They have been in your family, passed mother to daughter without interruption, for longer than the oldest monument on the planet has existed. Whatever haplogroup you carry, and if you have done a genetic ancestry test, and this information is available in your results, it traces back through the same maternal chain, to the same geography, to the same events, to women whose names were never recorded, and whose faces we cannot reconstruct, but whose biology we can now read with enough precision to understand what they were doing that kept the lineage alive. The genetic record does not preserve their names, but it preserves the consequence of their choices. That consequence is you. To understand what those women survived, the scale of the environmental crisis needs to be stated in physical terms that resist the tendency of geological language to abstract away the human reality. Between approximately 135,000 and 75,000 years ago, the tropical regions of Africa experienced a series of mega-droughts that represent some of the most severe climate events documented in the geological archive for the past 500,000 years. This is not a comparison to modern drought conditions or to the periodic rainfall deficits that modern meteorological systems classify as drought emergencies. The events documented in the lake sediment record are of an entirely different order. Research analyzing sediment cores extracted from Lake Malawi in East Africa found that during the worst episodes within this window, the lake's total water volume was reduced by at least 95%. Lake Malawi today is more than 500 meters deep, more than 570 kilometers long, and contains approximately 8,000 cubic kilometers of fresh water. A 95% reduction in the water volume of a lake of that size, sustained across the decades and centuries that the sediment record indicates, requires aridity so extreme and so prolonged that the surrounding landscape would have been transformed from diverse, a productive tropical ecosystem to something approaching near desert conditions across a region spanning thousands of kilometers. The word drought is inadequate for what the sediment record describes. What it describes is the systematic withdrawal of the conditions that make most of Africa livable. This paleoclimate record correlates directly with a genetic signal that is visible in the mitochondrial DNA of every living human being. Human mitochondrial DNA diversity is notably and distinctively low compared to what population genetics would predict for a species of our age and geographic range. A chimpanzee population, despite being far smaller in total number than the human global population, carries substantially more mitochondrial genetic diversity than humans do, because chimpanzee populations have not been compressed through the kind of bottleneck events that the human genetic record preserves. The simplest explanation for human mitochondrial genetic poverty is serial bottleneck events, repeated catastrophic reductions in population size that eliminated most genetic lineages, followed by expansion from a small surviving founding group whose limited genetic starting material became the foundation of all subsequent human genetic diversity. The timing of the most significant bottleneck signals in the mitochondrial clock aligns with the timing of the most severe documented climate episodes in the African paleoclimate record. The climate data and the genetic data are describing the same events from different angles. One angle is the physical world. The other is the surviving population's biological memory of what that world did to them. The supervolcanic eruption of Mount Toba in what is now Sumatra, Indonesia, approximately 74,000 years ago, represents an additional and well-documented stress event superimposed on a population already under sustained pressure from the preceding mega-drought cycle. Toba was among the largest eruptions in the past 2 million years of Earth history. It ejected an estimated 2,800 cubic kilometers of volcanic material into the atmosphere, roughly 3,000 times the eruptive volume of the 1980 Mount St. Helens event, and larger than any eruption that has occurred in recorded human history. The ash fall from Toba was extensive enough to deposit a layer approximately 5 centimeters deep across the entire Indian subcontinent, a geographic area of over 4 million square kilometers. Volcanic winter conditions following an eruption of this scale, the injection of sulfur aerosols into the stratosphere blocking solar radiation for years, would have produced measurable temperature reductions across Africa, Asia, and Europe, and would have severely disrupted the plant growth on which terrestrial food chains depend. Archaeological and genetic evidence regarding the magnitude of the human population impact from Toba specifically remains scientifically contested. What is not contested is that its timing falls within a period of already serious human population stress, and that whatever its independent contribution, it was experienced by a species that was not in a position of robust population health when the eruption occurred. The refugeia that preserved human populations through the mega-drought episodes appear to have been concentrated on coastlines, with the southern African coast providing the most archaeologically documented evidence of sustained occupation across the full duration of the crisis period. The physical characteristics that made this specific coastline survivable when inland Africa was not. Year-round shellfish resources producing consistent, reliable protein calories in a geographic zone that the mega-droughts, whose impact was primarily on inland freshwater and vegetation, could not fully deplete. Marine shellfish beds are maintained by oceanic upwelling systems that were not directly affected by the terrestrial aridity characterizing the mega-drought episodes. Cave systems cut into the coastal cliffs by wave action over millennia provided shelter that maintained livable temperature ranges even when the exposed landscape outside was climatically marginal. Freshwater springs emerging at the base of the sea cliffs in several locations along the southern Cape coastline provided drinking water in proximity to the marine food resources. The site was not comfortable. No site available to a small, technologically limited human group during this period was comfortable, but it was survivable, and it remained survivable across a period during which most of Africa was not. The people maintaining occupation of sites like Pinnacle Point across this period were not passively enduring the climate. They were actively navigating it, making decisions about where to go and when, to reading the seasonal and annual signals that indicated whether the inland food sources were recoverable or required abandonment for another season, managing the social and logistical challenges of sustaining a group of 25 to 50 people in a resource environment that required daily, detailed expert knowledge of where the calories were. The ochre workshops and the symbolic markings at Blombos Cave are not incidental to this survival story. They are evidence that the cognitive architecture required to maintain the cultural transmission of knowledge, the organizational capacity to run a workshop, to train others in a process, to use symbolic marking as a memory or communication tool, was fully operational in the people living through the worst of these events. They were not cognitively diminished by stress. They were fully modern minds, deploying their full modern cognitive capacities under conditions that demanded everything those capacities could provide. Wherever you are right now as you listen or read, wherever in the world your particular night or morning finds you, the mitochondrial record draws a direct line from your location to this coastline. The oldest maternal lineages in every human population on Earth trace back through the same filter, the same bottleneck, the same compressed surviving group. Whether your ancestry is West African or East Asian or Northern European or Indigenous American, your maternal line passed through the same genetic eye of the needle. The populations that eventually spread to populate every continent on the planet expanded from the surviving coastal populations of this period. The diversity of all 8 billion living humans traces back to the genetic starting material that existed in a population that may have been smaller than the attendance at a large university football game. The women who held those coastal groups together through the worst of the climate events were not chosen by any conscious selection process. They were the ones who happened to be alive when the crisis reached its peak, who happened to possess the specific combination of ecological knowledge and social intelligence that kept their group's caloric balance positive through the worst episodes, and whose groups therefore survived to pass their genetic signatures forward. The oldest women among them, the ones who had been accumulating specific, tested, location-precise knowledge of this coastline for the longest time, were doing something that the genetic record now allows us to describe as evolutionarily consequential. But there is a deeper question this chapter leaves unanswered. The women who survived were not just knowledgeable. Their biology was configured differently from younger women in a way that made that knowledge more available, more deployable, more productively directed toward the group's survival. Understanding that biological configuration requires examining what menopause actually does, not what it takes away, but what it changes the organism into. Menopause presents an immediate paradox to anyone who thinks carefully about evolutionary logic, and the paradox deserves to be stated without the hedging that typically softens it in popular accounts, because the unhedged version is the one that makes the answer, when it arrives, genuinely surprising. Natural selection maximizes reproductive output. This is not a rule with numerous exceptions, or a principle that holds only under certain conditions. It is the fundamental mechanism by which gene frequencies change across generations, the single most consistently supported principle in the entire body of evolutionary biology. An individual who produces more surviving offspring than competitors passes more copies of her genes to the next generation. Genes that increase reproductive output spread. Genes that decrease it are eliminated. Over sufficient time and sufficient generations, that this process produces organisms that are very good at reproducing, which is why the living world is populated by organisms that, with only rare exceptions, reproduce until they cannot. A 50-year-old female chimpanzee, if she survives that long in the wild, is still attempting to reproduce. Reproduction does not stop on schedule. It continues until the biological capacity for it is exhausted, and then the animal dies relatively soon after. This pattern characterizes the overwhelming majority of mammalian species. Menopause, as it occurs in humans, violates it completely. The biological machinery shuts down in midlife. The individual survives for decades more. The genes encoding this shutdown were selected for, accumulated in hundreds of contributing variants, rather than selected against. The question of why requires taking the paradox seriously, rather than explaining it away. The first formal evolutionary explanation for menopause came through what researchers refer to as the lifespan artifact hypothesis, or sometimes the mutation accumulation hypothesis in its more technical formulations. The argument runs as follows. The human female ovarian reserve, approximately 1 million follicles present at birth, declining continuously through cell death throughout life, was calibrated by ancestral selection to last approximately as long as the ancestral female lifespan. In environments where adult mortality was high and few women survived past 50, the follicle reserve simply ran out, around the time most women were dying anyway. Selection had no opportunity to extend the follicular reserve because there were no post-menopausal women present in sufficient numbers to select for longer reproductive capacity. Menopause, on this view, is not an adaptation at all. It is an artifact, a biological schedule calibrated to a life expectancy that no longer applies now that modern medicine has extended survival well past the point where evolution calibrated the ovarian clock to stop. This hypothesis has an appealing simplicity, and it was the dominant explanation in evolutionary medicine. For long enough that many people who learned about menopause in medical or biological training absorbed it as settled fact. The problem is that it does not survive contact with the actual mortality data from pre-industrial and pre-modern human populations. Detailed analyses of age-specific mortality patterns in populations without access to modern medicine, including historical demographic records from rural Gambia, pre-industrial Finland, and 18th century Canada, as well as skeletal age-at-death analysis from archaeological cemetery populations across multiple regions and time periods, consistently show that women in traditional societies regularly survived well past 50 years of age. A 45-year-old woman in a functional hunter-gatherer group with access to adequate food had a reasonable probability of living another 20 to 30 years. Post-menopausal women were not a modern invention. They were present in ancestral populations, in numbers significant enough to be behaviorally and ecologically consequential, which means selection did have the opportunity to act on post-menopausal life history, and the absence of selection for extended fertility in post-menopausal women requires an active explanation, not a passive one. This is where the scientific debate becomes genuinely interesting, because the two primary alternative explanations each capture something true, while each leaving something unresolved. The grandmother hypothesis, developed most extensively by University of Utah anthropologist Kristen Hawkes and her colleagues across a body of work spanning 3 decades, argues that menopause is an active adaptation, specifically that stopping reproduction at approximately 50 years of age increases a woman's inclusive fitness by allowing her to redirect the caloric, physical, and cognitive resources previously invested in reproduction toward the survival and nutritional support of grandchildren. Because grandchildren share approximately 1/4 of a grandmother's genes, the inclusive fitness accounting can make grandmothering a more reproductively efficient use of a postmenopausal woman's resources than attempting a late-life pregnancy. If the grandchildren's survival probability is meaningfully improved by the grandmother's contribution, and if the risk of the late-life pregnancy to the grandmother herself is significant. Hawkes's fieldwork among the Hodza of Tanzania provided the first systematic empirical evidence that this fitness accounting actually works in a traditional foraging population, documenting measurable differences in grandchild nutritional status and survival rates correlated with grandmother proximity and activity levels. The competing hypothesis, associated with researchers including Jan Weisse and Eckhart Voland, who analyzed historical Finnish and Canadian parish records, emphasizes the mortality risk of late-life reproduction as the primary selective pressure. Childbirth under ancestral conditions was not a medically managed event with controlled infection risk, surgical intervention capacity, and blood transfusion availability. It was a physiologically demanding process with a mortality risk that was non-trivial across all maternal age groups, and that increased with advancing maternal age as the physiological reserves required for successful labor diminished. Analysis of historical demographic data from pre-industrial European populations found that women whose mothers stopped reproducing earlier, who did not attempt late-life pregnancies, had higher survival rates themselves because their mothers survived longer into their own old age and provided more sustained assistance across the child-rearing years. The mortality risk of maternal late-life reproduction was partially borne not just by the mother, but by her existing children who lost their mother's contribution to their survival when a late-life pregnancy killed her. Stopping early protected existing investments. Both hypotheses have supporting evidence. Both have specific weaknesses. The grandmother hypothesis faces challenges from demographic modeling, showing that the fitness gains from grandmothering under the most realistic assumptions about ancestral group size, kinship structure, and the actual magnitude of grandmothering's caloric contribution may not always be large enough to overcome the direct reproductive cost of stopping, particularly in the early generations of the transition, before post-reproductive longevity had been selected to its full modern extent. And the stopping early hypothesis cannot fully account for the active, energetically substantial investment of post-reproductive women and grandchildren documented across multiple traditional societies. If stopping early were the only advantage, selection would favor simply dying after the last pregnancy rather than maintaining decades of additional metabolically costly survival. The current state of scientific consensus is not a clean victory for either position. It is the recognition, supported by the most recent theoretical modeling, that both pressures operated simultaneously, pointing in the same direction, and that the 290 variant genetic architecture of menopause timing reflects the convergence of multiple selection pressures on the same biological outcome. What the debate between these hypotheses has not fully addressed is the biological mechanism of the transition. When estrogen declines at menopause, bone resorption accelerates in the trabecular bone of the spine and hip by enough to produce measurable bone density losses within the first years post-menopause. Cardiovascular risk profiles change as the direct vascular protective effects of circulating estrogen on arterial wall integrity are removed. Sleep architecture is reorganized in ways that affect slow-wave sleep depth and alter the 24-hour cortisol distribution. Body composition redistributes visceral fat in patterns associated with changed metabolic signaling. These are not the changes of passive depletion, not a battery that has run down. They are the changes of coordinated multi-system reorganization occurring simultaneously across different organ systems in response to a shared hormonal signal. The body is not stopping, it is converting. The question of what it is converting into, what the post-menopausal biological system is configured to do, that the reproductive system was not, is the question that the next chapter answers with evidence from 3 independent sources. The unresolved question from the previous chapter requires an answer drawn from 3 independent evidence streams, because a single source would leave the conclusion insufficiently supported for the weight it carries. The claim to be established is this: the biological costs visible in the menopausal transition are not evidence of deterioration or passive rundown. They are the signature of a major resource reallocation operating simultaneously across multiple biological systems. The body is changing what it does with the resources previously allocated to maintaining reproductive readiness. That reallocation was adaptive under the conditions that produced it. The evidence for this interpretation comes from caloric biology, from systematic behavioral observation of traditional forager populations, and from mathematical modeling of life history evolution. and all three sources converge on the same conclusion from different angles of approach. The caloric evidence begins with basic reproductive biology. Maintaining the human female reproductive cycle across the full span of the reproductive years is not a metabolically trivial investment. The hormonal infrastructure of the cycle, follicle-stimulating hormone release, follicular development, the LH surge driving ovulation, the luteal phase maintenance of the uterine environment in preparation for potential implantation, represents a continuous monthly investment of metabolic resources, whether or not conception occurs. Pregnancy and lactation together require an additional 500 to 550 calories per day, above baseline metabolic needs, sustained across the approximately 18 months of pregnancy plus the lactation period that typically follows. The follicular phase itself, and with its intensive hormonal preparation, requires measurable glucose and lipid allocation from the body's daily energy budget. When reproduction stops, these caloric investments do not disappear. The food being consumed continues to be consumed. The metabolic capacity continues to operate. The calories that were previously allocated to reproductive infrastructure are redirected to other functions. The post-menopausal body is not calorie poor relative to its reproductive age state, for it is calorie redirected. The question is where those calories go, and whether the destination represents an adaptive use of the resource. The behavioral evidence comes from Kristen Hawkes's landmark fieldwork among the Hodza of north-central Tanzania, conducted over multiple field seasons beginning in the 1980s and generating a series of foundational papers that established the empirical basis of the grandmother hypothesis. The Hodza are a small-scale forager society, one of only a handful remaining on Earth, in which caloric acquisition through daily foraging remains the primary basis of subsistence. Hawkes and her colleagues conducted systematic time allocation and food production studies, observing and recording the daily activities of Hodza individuals across sex and age categories with enough consistency to permit quantitative comparison. And the finding that anchored the grandmother hypothesis was this: post-menopausal Hodza, women forage more hours per day and produce more food per hour of foraging effort than reproductive age women with dependent children. The difference in productive output between these two groups is not marginal. Reproductive age women with infants or young children spend substantial portions of each day in direct child care that structurally limits their foraging range and their total daily productive output. Post-menopausal women carry none of this constraint. They wake early, move through the foraging landscape at their own pace and to the limits of their physical range, and return with food in quantities that consistently exceed their own individual caloric needs. The surplus does not accumulate. It flows to grandchildren, specifically to the children of daughters who are occupied with younger siblings and whose own food production capacity is therefore structurally limited by the same infant care constraint that limited their own mothers at the same life stage. The caloric contribution of Hodza grandmothers to their daughters' children is measurable in ways that have direct consequences. When Hodza children's nutritional status is analyzed in relation to the presence or absence of a grandmother in the same residential group, children with actively foraging grandmothers show measurably better growth and nutritional indices than children without grandmother proximity. The relationship is not explained by genetic quality differences between the families involved, or by the economic circumstances of the fathers, or by other confounding variables that researchers have tested. It is specifically the grandmother's foraging contribution that accounts for the difference. The grandmother effect in Hodza populations is a caloric fact with survival implications. It is not a cultural sentiment about the value of older women. It is a measurable contribution to a measurable outcome in the next generation's health. A mathematical model published in 2014 in the Journal of Theoretical Biology by Pia Smaldino, James Coxworth, James McQueen, and Kristen Hawkes used computer simulation to test whether this kind of grandmothering behavior alone, under realistic demographic assumptions, could drive the evolution of extended post-reproductive longevity, starting from a chimpanzee-like life history, baseline. The simulation began with a virtual population whose females reproduced until late life and died relatively quickly after their last offspring, modeling the life history pattern of our closest living relative. Grandmothering behavior was then introduced. Post-reproductive females provisioned grandchildren with caloric surplus from their foraging activity. The simulation ran across multiple thousands of simulated years. The result was striking. Average adult lifespan in the simulated population extended from approximately 25 years to over 49 years within 37,000 simulated years of selection. 37,000 years is a brief span in evolutionary terms, and it represents roughly 1,500 human generations. The simulation found that grandmothering behavior alone, without any additional mechanisms, was sufficient to drive the evolution of substantially extended post-reproductive longevity under realistic assumptions about the magnitude of the fitness benefits involved. This does not prove that this is exactly what happened in human evolution. It demonstrates that the mechanism is capable of producing the outcome, that the arithmetic works. The neurological evidence for the redirection interpretation comes from a different direction entirely, and it is the evidence that most directly addresses the lived experience of the menopausal transition. If menopause were simply biological deterioration, a system running down without purpose, cognitive function should decline continuously and irreversibly from the onset of perimenopause onward. Continuous deterioration is what breakdown looks like. And a 2021 study published in Nature Scientific Reports, led by Lisa Mosconi of Weill Cornell Medicine, used structural and functional neuroimaging to track brain changes across the menopausal transition in a cohort of women at different transition stages. The finding: brain gray matter volume and connectivity did change during the active transition phase, but post-menopause, both measures showed recovery and stabilization. Cognitive performance was preserved in postmenopausal women, and the preservation correlated specifically with recovery of brain adenosine triphosphate production, the cellular energy currency. The brain was not deteriorating through menopause. It was reorganizing, temporarily reducing in some functional metrics during the active transition, and then stabilizing into a different but not diminished functional configuration, post-transition. The fog is real. The destination of the fog is not deficit, it is difference. If you are in perimenopause now, or have been recently, or are watching this because someone you love is navigating the transition, the difficulty is not imaginary. The sleeplessness is not psychosomatic. The cognitive changes of perimenopause are neurologically real, with a measurable physiological basis in the changing glucose metabolism of specific brain regions, and they are the experience of a biological system undergoing a significant reorganization, not breaking down, but converting from one functional configuration to another. The discomfort is the cost of the conversion. The converted state is not lesser. It is different and differently capable, And those different capabilities were specifically useful to groups of women who had stopped reproducing and taken up a role in the survival of the group that reproduction had not permitted. What that role required in terms of specific knowledge and cognitive capacity is the question that the chapter following this one was built to answer. The woman walks the shoreline in the gray light before sunrise, and she is moving with the specific kind of purposefulness that belongs only to someone who does not need to look around. She knows this coast the way you know the rooms of a house you have lived in for decades, not by consulting a stored description of the layout, but by having internalized the layout so thoroughly that navigating it requires no conscious attention, leaving the conscious mind free to do other things. She is 51 years old. She last carried a child 7 years ago, and the child survived. What she carries now, instead of an infant, is a cognitive map of this coastline spanning 3 decades of daily observation, continuously tested against the actual outcomes of the decisions it guided, and continuously refined by the corrections those tests required. She knows that the tidal flat 2 kilometers north, which yields more shellfish per square meter than any other site within the group's regular range, shifts its most productive zone approximately 20 meters south each winter, as the offshore current changes direction with the seasonal wind pattern shift. She knows this because she has observed it over many winters, not heard it described. She knows that this winter's shift has not yet occurred, because the wind pattern has been unusual for the past 3 weeks, and she has been watching it. She knows which of the young women in the group has the most developed skill for reading the tide timing and could be trusted to manage a collection party without her supervision, and which of the young men tends to harvest too aggressively from the most productive sites in ways that will reduce yield over the following weeks. She manages all of this information simultaneously, without apparent effort, because it has been organized by decades of experience into something that no longer feels like knowledge. It feels like perception. Researchers studying contemporary forager societies have documented the depth and specificity of ecological knowledge held by older women in multiple ethnographic contexts across several continents. The documentation comes not from asking older women to describe what they know, a method that tends to produce the conscious, articulable portion of knowledge rather than the full body of it, but from observing their foraging behavior across seasons and years and comparing outcomes. Among the Hodza of Tanzania, post-menopausal women demonstrate detailed knowledge of underground tuber locations that researchers have confirmed through GPS mapping and seasonal return visit tracking. The knowledge is not general. Not tubers are found in clayey soil near seasonal water, but specific, location-precise, all and seasonally calibrated to a degree that takes years to acquire even under direct instruction. Experienced Hodza women navigate directly to specific tuber locations across hundreds of meters of undifferentiated-looking savanna, excavate at a specific spot rather than conducting a broad search, and reach productive yield with a consistency that is not replicable by observers who do not share their location, specific experiential database. When older women are absent from foraging groups, the foraging productivity of the remaining members declines measurably because what the older women know is not fully transferable to someone who has not spent the same years acquiring it in the same landscape. The parallel in the ocean makes the mechanism visible in ways that ethnographic observation of human societies cannot achieve, because the mechanism has been studied in enough quantitative detail over enough years to establish causation rather than correlation. Research by Darren Croft on Samuel Ellis and colleagues at the University of Exeter and the University of York, drawing on 40 years of behavioral and demographic data from the southern resident killer whale population compiled by the Center for Whale Research, has quantified what happens when a post-reproductive matriarch is removed from a killer whale family. When a grandmother orca dies, her grandchildren are approximately 4.5 times more likely to die in the following 2 years than peers whose grandmothers remain alive. Her adult sons, mature males capable of hunting independently in the prime of their physical strength, face a 13-fold increase in their annual mortality risk following their mother's death, compared to periods when she was alive. These are not marginal effects of no practical consequence. They represent the difference between a lineage that persists through the next decade and one that effectively ends within 2 years of losing its matriarch. The magnitude of the post-reproductive female's contribution to her descendants' survival, measured in killer whales with a precision that human populations cannot provide, suggests that the grandmother effect is not a marginal supplement to baseline survival. It is a structural component of how these groups function, and its removal produces an immediate demographic collapse. The primary mechanism identified in the killer whale research is ecological knowledge, specifically the knowledge of where salmon foraging grounds are located during years when salmon abundance is reduced. Killer whales of the resident ecotype are dietary specialists. They eat salmon, and salmon almost exclusively. The salmon runs on which they depend shift annually in their spatial distribution along the Pacific coastline, with the magnitude of the shift varying by year in ways that are not predictable from simple observation of current conditions. During years of reduced salmon abundance, the difference between finding the fish and not finding them is the difference between adequate nutrition and starvation for the entire pod. Post-reproductive female orcas preferentially lead their pods toward salmon during exactly these low abundance years. And the ecological knowledge they carry, the multi-decade experiential database of where the fish go when conditions change, is most valuable and most irreplaceable precisely when conditions are most stressful and the fish are hardest to find. The grandmother matters most during the crisis, the parallel to the prehistoric coastal woman who knows where the shellfish beds shifted during the last drought year is not metaphorical. It is mechanistic. The social and political dimension of knowledge held by older women in small groups is harder to quantify than caloric contribution or ecological navigation, but it is not less real. The oldest women in a band of 25 to 50 people carry the social memory of the group across a longer span than anyone else in the group. They remember which families shared food without being asked during the shortage 3 winters ago, and which did not. They remember the terms of the relationship with the coastal band 20 kilometers north, who in that group was reliable in conflict, who was not, and what the basis of the relationship established between the groups during the severe drought 8 years ago was, when the 2 groups shared territory for 2 seasons out of necessity. They remember which plants caused illness in the season that their own mother's mother showed them, and which preparation technique made them safe. But this information is not written down and cannot be looked up. It exists only in the memories of the people who were present when the events occurred. When the oldest woman in the group dies, everything she remembers that no one else was old enough to witness disappears with her. No reconstruction is possible from descriptions, because the descriptions do not exist. The knowledge was never put into the form of conscious description. It lived as experience. Tell us about the oldest woman you have known, your grandmother or your mother's closest friend, or a neighbor who had been in the same place for 50 years when you were young. Think about the specific things she knew, not the things she told you she knew, but the things that became apparent in moments of difficulty or decision when information you didn't know you needed turned out to be in her possession. The category of knowledge that is invisible until it is needed and irreplaceable once it is lost. Because what the science of why you exist is describing, at its most specific level, is exactly that category of knowledge, held in exactly that kind of person, at exactly the moment in history when it determined whether an entire lineage survived or not. The biological argument for menopause as an evolutionary adaptation achieves its greatest evidential strength when examined not in human populations alone, but through the comparative biology of the 5 whale species that arrived at the same solution through entirely independent evolutionary paths. Human evolutionary history, as a subject of study, suffers from an unavoidable limitation. We have no control condition. There is no population of anatomically modern humans who did not evolve menopause against which to compare those who did. The evolutionary experiment that produced menopause ran once in the human lineage, and we are all descendants of the result. We can study the genetics of the outcome, the paleoclimate of the period during which selection operated, and in the behavioral ecology of living populations that still reflect the social structures within which selection operated. But we cannot directly observe what human groups without post-reproductive matriarchs would have looked like in the environments where menopause evolved. The killer whale data solves this problem by providing an independent replication of the evolutionary experiment conducted under different ecological conditions in a completely different biological lineage, with outcomes quantified across 4 decades of individual-level longitudinal tracking that no human population study has ever achieved. A comprehensive analysis published in the journal Nature in March 2024 examined the life history characteristics of 32 species of toothed whales, covering the full range of cetacean species for which adequate demographic data was available. The researchers compared reproductive lifespan, total lifespan, to run the ratio of post-reproductive to reproductive years across menopausal and non-menopausal species. The 5 menopausal species, killer whales, short-finned pilot whales, false killer whales, narwhals, and belugas, share a distinctive life history profile that none of the 27 non-menopausal species in the comparison exhibit. The menopausal species live dramatically longer than their reproductive period alone would predict, with post-reproductive lifespans representing substantial fractions of their total adult lives. Female killer whales stop reproducing in their late 30s to early 40s. Their maximum documented lifespan is approximately 90 years. The difference between the age at last reproduction and the maximum lifespan is more than 50 years. More than 50 years of biologically active, socially integrated, ecologically productive life after the last calf. The post-reproductive period, measured as a proportion of total adult life expectancy, represents more than 30% of the killer whale female's adult years. In the southern resident killer whale population specifically, where individual identification records have been maintained continuously since 1976, females can expect to live at least 16 to 20 years after their last reproductive event, a post-reproductive window longer than the entire adult life of many mammalian species. And the non-menopausal toothed whale species show a starkly different pattern. They reproduce until late in life and die relatively shortly after reproductive capacity is exhausted. The pattern expected by straightforward reproductive senescence in the absence of grandmothering selection. Critically, the 2024 Nature analysis found that menopausal and non-menopausal species do not differ meaningfully in the total length of their reproductive periods. Both groups spend roughly comparable spans of years in reproductive activity. The difference is entirely in total lifespan. The menopausal species have longer lives, and all of the additional life accumulates in the post-reproductive phase. Evolution did not shorten the reproductive period of menopausal species. It extended their total lives, building all of the extension at the back end in the years after reproduction has ceased. The selection pressure was for longer post-reproductive life, not for earlier reproductive cessation. Menopause in toothed whales did not arise once, from a single ancestral event, and spread through inheritance. The last common ancestor between humans and any cetacean lived approximately 90 million years ago, long before either lineage had developed the social complexity, the large brain, or the ecological dependence on accumulated knowledge that appears to underlie the grandmother effect in both groups. Within the toothed whale lineage itself, the trait arose independently at least 4 separate times, in lineages that diverged from each other over millions of years, that occupy different geographic ranges, and that specialize in different prey species and different ecological contexts. Killer whales and narwhals are not closely related within the cetacean tree. They share menopause not because they share a recent common ancestor who had it, but because they independently arrived at the same biological solution to the same selection pressure. When evolution produces the same outcome in multiple independent lineages that share no recent common ancestor, the explanation is convergence. The same selective pressure operating in different biological contexts and arriving at the same answer, because the answer is genuinely better than the alternatives under those specific conditions. The quantification of the grandmother effect in killer whales is specific enough to describe mechanistically, rather than correlatively. Work by Nattrass and colleagues at the University of Exeter, published in PNAS in 2019, established that the survival benefits provided by post-reproductive female killer whales to their offspring and grandchildren are large enough, under realistic demographic assumptions, to explain the evolution of extended post-reproductive longevity through inclusive fitness accounting. Grandmothering is worth the reproductive cost of stopping. The indirect fitness gains, through improved survival of relatives who share the grandmother's genes, exceed the direct fitness loss of not producing additional offspring. This is the foundational fitness calculation that the grandmother hypothesis requires, and in killer whales it has been demonstrated with enough rigor to satisfy the skeptics within the field. A post-reproductive female orca is not simply surviving past her useful biological date. She is doing something specific, with measurable consequences, for the survival of individuals who carry her genes. Her post-reproductive existence is not biological inertia. It is biological function. The debate that remains productive within the field concerns the precise relative contributions of different grandmother functions to the overall survival benefit. The leading candidate mechanism is ecological knowledge, the post-reproductive female's multi-decade database of where food can be found under different oceanographic conditions, most valuable and most irreplaceable precisely during periods of low prey abundance. Additional mechanisms include direct food sharing with adult sons, a behavior documented in the southern resident killer whale data, showing that post-reproductive mothers preferentially share salmon with their adult male offspring in ways that reduce those males' vulnerability to nutritional stress during lean salmon years. A third mechanism, documented in a current biology paper by Grimes and colleagues in 2023, involves social conflict reduction. The presence of a post-reproductive mother reduces the frequency of socially inflicted toothache injuries to her sons from other males in the group, suggesting that the matriarch's social authority or conflict management behavior provides protection to her adult male descendants from within-group social aggression. The grandmother is simultaneously the navigator, the food sharer, and the social regulator, a multifunctional contribution to group function that no single mechanism fully captures. For prehistoric human women, the knowledge held by post-reproductive matriarchs was different in specific content. No salmon runs, no oceanographic memory, but identical in structural function. It was multi-decade, location-specific, failure-tested environmental intelligence whose value was greatest during resource scarcity, and combined with social and political memory, spanning the longest available time horizon in the group, combined with the caloric contribution of a highly productive forager, unconstrained by infant care. The whale data provides the controlled comparison that human evolutionary history cannot provide. It shows, with quantitative precision, that the grandmother effect is real, that its magnitude is large enough to drive evolutionary change, and that it has been independently discovered by evolution at least 5 times across the history of mammalian life. The human case is not unique. It is the most studied case of a broader phenomenon, whose independent occurrences constitute the most powerful available evidence that post-reproductive matriarchs were genuinely, consequentially valuable to the groups they remained part of. A paradox stands at the center of the relationship between menopause and cognition, and its resolution changes how the menopausal transition can be understood by the people who navigate it. The paradox is this: if the menopausal transition is primarily a biological deterioration, a system winding down, a body past its evolutionary prime gradually losing function, cognitive performance should decline continuously and irreversibly from the moment perimenopause begins, because deterioration is continuous and progressive by definition. But the neurological evidence from the past decade does not show continuous progressive cognitive decline through the menopausal transition. It shows something more complex and, in its implications, more interesting. Understanding what that evidence shows requires a brief examination of how estrogen interacts with brain function, because the interaction is more specific and more consequential than most people navigating the transition have been told. Estrogen plays a significant and well-documented role in the brain that extends well beyond reproductive function. Estrogen receptors are present in multiple brain regions, including the prefrontal cortex, the hippocampus, and regions associated with social cognition and emotional processing. One of estrogen's principal brain functions is the facilitation of cerebral glucose metabolism, the efficiency with which brain cells take up and use glucose as their primary fuel. When estrogen levels are high, cerebral glucose uptake is efficient and consistent across the brain regions where estrogen receptors are concentrated. When estrogen declines, as it does during the perimenopause and early post-menopause period, the efficiency of glucose uptake in those regions temporarily decreases. So the brain continues to function, but the fuel processing becomes less efficient, like an engine operating on a slightly different fuel mixture than it was optimized for. The subjective experience of this metabolic shift is the cognitive fog that women consistently report during perimenopause, the word that doesn't come quickly, the concentration that slips, the sense of a mind that is functioning but working harder than usual for the same outputs. This experience has a specific neurological basis. It is not psychosomatic, and it is not imaginary. The critical finding in the neurological research of the past decade is what happens after the active transition. A 2021 study led by Lisa Mosconi at Weill Cornell Medicine used structural MRI, functional neuroimaging, and brain metabolic assessment to track brain changes longitudinally across groups of women at different stages of the menopausal transition: premenopausal, perimenopausal, and post-menopausal. The findings distinguished between what happens during the transition and what characterizes the post-transition state. During perimenopause, brain gray matter volume decreased in some regions, and measures of functional connectivity changed. These are the changes underlying the cognitive symptoms of the transition. But in the post-menopausal women, those who had completed the transition rather than being actively in it, both gray matter volume and functional connectivity showed evidence of recovery and restabilization. Brain adenosine triphosphate production, the measure of cellular energy generation in neural tissue, recovered post-menopause in ways that correlated directly with preserved cognitive performance. The brain was not completing a progressive deterioration, so it was completing a transition from one metabolic configuration to another. The comparison to a fuel switch is mechanistically accurate as well as descriptively useful. When the brain's primary fuel processing system recalibrates from estrogen-facilitated glucose metabolism to a post-menopausal metabolic baseline, it goes through a period of reduced efficiency during the recalibration. The efficiency reduction is the cognitive fog. The completion of recalibration is the restoration of function, not to the identical premenopausal profile, but to a stable, functional, well-maintained post-menopausal profile. The two profiles are different. Post-menopausal brain function is not reproductively oriented in the same way. The hormonal modulation of cognition that served the reproductive phase of life is absent. What remains is a cognitive system operating at the metabolic baseline the brain has now calibrated for. With all the experiential knowledge accumulated across decades of learning intact, and with the social cognition and pattern recognition functions that appear particularly durable through the transition available at full capacity. Research on the specific cognitive domains most and least affected by the menopausal transition produces a consistent pattern across multiple study populations. Verbal fluency and narrative memory, the ability to recall and organize connected sequences of information, remain robust in healthy post-menopausal women across multiple subsequent decades. Processing speed for certain types of novel information shows modest age-related changes that are not unique to the postmenopausal period and are not attributable to menopause specifically when age is properly controlled. And social cognition, the multidimensional capacity to accurately read emotional states in others, interpret the dynamics of social groups, anticipate how individuals will behave under different conditions, and navigate complex social situations involving multiple parties with different motivations, shows no characteristic postmenopausal decline in healthy women. Some studies find that experienced older women outperform younger women on complex social judgment tasks that require not just theory of mind capacity, but the kind of pattern recognition across social situations that only accumulated experience can provide. This cognitive profile, durable verbal and narrative memory, robust social cognition, pattern recognition, drawing on decades of experience, describes precisely the cognitive toolkit most valuable to a woman whose role in an ancestral group had shifted from reproduction to the provision of ecological knowledge, social mediation, and intergenerational guidance. This alignment is not coincidental in any trivial sense. The same selection pressures that shape the timing of menopause, operating across the same bottleneck events in the same compressed populations over the same tens of thousands of years. We're also operating on the post-menopausal cognitive profile. The body that emerged from reproduction was being shaped by selection towards specific functional capabilities, not abandoned by selection as past its useful life. The menopausal brain is not an evolutionary afterthought. It is an evolutionary product, shaped by the same forces and toward the same ends as everything else the selection pressure of that period produced. Upon your own experience of cognitive change during the menopausal transition, the specific difficulty of perimenopause, the particular quality of the postmenopausal mental landscape for women who have passed through the full transition, is the surface of a neurological reorganization whose depth is 150,000 years of selection. The reorganization was built by the same women, on the same coastlines, under the same conditions that this documentary has been describing. And the fog is not failure, it is the cost of the conversion. The destination of the conversion, a cognitive system configured for the specific functions that kept the lineage alive during every crisis that generation faced, is not lesser than the starting point. It is different and differently suited to the second half of a life in which experience has accumulated to the depth that makes it genuinely useful. The scope of the claim this documentary has been building toward requires clear statement before the convergence of evidence that substantiates it is assembled, because claims of this magnitude deserve to be held accountable to the evidence that supports them rather than softened into generalities. The claim is this: menopause, the most physically significant hormonal transition in the human female life course, by involving years of biological disruption and a permanent reorganization of physiological function, persisted in the human genome because it was adaptive, because the adaptiveness was sufficiently large and sufficiently consistent across sufficient generations of catastrophic environmental pressure to accumulate 290 measurable genetic variants in the modern human genome. And because the conditions that generated this selection pressure were specific enough that they can be identified in 3 independent scientific records whose contents, examined separately, each point toward the same conclusion. The 3 records are the genetic record preserved in living human DNA, the environmental record preserved in African lake sediment cores and other paleoclimate proxies, and the behavioral and demographic record accumulated across 40 years of killer whale observation and multiple ethnographic studies of traditional forager populations. Each record is independently maintained, independently interpreted, and independently analyzed. All 3 point the same direction. The genetic evidence has been assembled across multiple sections of this documentary, but its full weight is most visible when the individual findings are held together. And the mitochondrial phylogeny shows that all living human maternal lineages trace back to a single ancestral population in sub-Saharan Africa, dated to approximately 150 to 200,000 years ago. The geographic distribution of the oldest surviving mitochondrial haplogroups clusters along the southern and eastern coasts of Africa, the same geography where the archaeological record places the coastal refugeia that preserved human populations through the megadrought episodes. The 2021 Genome-Wide Association study identified 290 genetic variants distributed across DNA repair pathways and reproductive hormone systems, contributing to the precise timing of menopause onset in ways that reflect not accident, but sustained polygenic selection. The 2016 PNAS analysis by Hawks and colleagues found genomic signatures consistent with selection for extended post-reproductive longevity in populations with the social structures predicted by the grandmother hypothesis. The genetic record, read in full, describes a population that passed through catastrophic bottlenecks, survived in coastal refugeia, and accumulated extensive polygenic selection for a specific life history feature, post-reproductive longevity, during exactly the period and in exactly the geography where the environmental record places the most severe survival pressure. The paleoclimate evidence is the record of the pressure itself. Lake sediment cores from Lake Malawi document at least 95% reductions in lake water volume during the mega drought episodes between 135,000 and 75,000 years ago. Similar evidence from Lake Tana in Ethiopia, where a 150,000-year paleoclimate record was recovered from sediment cores drilled close to early modern human fossil sites, shows repeated alternation between humid and severely arid periods, with the arid episodes sufficient in severity and duration to fundamentally disrupt the food chains supporting inland human populations. The Toba eruption at 74,000 years ago introduced an additional acute stress event into a population already operating under chronic resource limitation. The cumulative effect of these events on a species whose survival required daily caloric acquisition by small, knowledge-dependent groups in a landscape that was episodically transforming from productive to near barren was exactly the kind of sustained, severe, multi-generational selection pressure that the genetic record shows was experienced. The climate data does not just provide context for the genetic data, it describes the mechanism through which the selection operated. Wherever in the world you are listening to this right now, whatever continent, whatever climate, whatever season surrounds you tonight, the maternal line you carry traces back through the same filter that the climate and genetic data are jointly describing. Whether your ancestry is from populations that spread to East Asia 50,000 years ago, or to Europe 40,000 years ago, or that remained in Africa and gave rise to the extraordinary genetic diversity of the African continent, can all maternal lines trace back through the bottleneck to the coastal survivors of the mega-drought period. The science here does not belong to any particular population or any particular culture. It belongs to every person whose maternal line has survived to the present, which is every person alive. Tell us where in the world you are, because the story of where all of us came from begins in the same place. And the distance between your location and that South African coastline is measured in generations, not in separation. The behavioral and ecological evidence, the third line, has accumulated across 4 decades of killer whale observation and multiple generations of forager ethnographic fieldwork, and its specific findings have been detailed in the preceding chapters of this documentary. What the third evidence stream contributes, at the level of convergence rather than individual finding, is the demonstration that the grandmother effect is not a hypothesis that has been proposed and not yet tested. It is an effect that has been tested in killer whales with enough statistical precision to establish mechanism rather than mere correlation, and that produces effects large enough, a 4.5 times increase in grandchild mortality when the grandmother dies, a 13-fold increase in adult son mortality to satisfy the inclusive fitness accounting that the grandmother hypothesis requires. The same effect has been identified with somewhat less statistical precision but consistent direction in multiple human populations from the Hodza of Tanzania to historical populations in Finland and Gambia. The behavioral record and the demographic record across species and across human populations to show the grandmother effect operating in the direction and at the approximate magnitude that the genetic and paleoclimate records predict it should have. One paradox remains unresolved as this third line of evidence converges with the first two. If the grandmother effect is this powerful, if post-reproductive matriarchs provide this much survival benefit to their descendants, why did only 6 species across all of mammalian life evolve the response of menopause? And of the approximately 6,000 mammalian species on Earth, why not 60 or 600? The conditions that produced this adaptation in humans and 5-toothed whale species should, if those conditions are common, have produced it in many more lineages. The fact that it appears in only 6 suggests that the conditions are not common, that they require a very specific combination of ecological, social, and demographic circumstances that almost never co-occur. What those circumstances are, stated precisely, is the answer that the final chapter of this documentary was built to provide. The paradox with which the previous chapter closed. The question of why only 6 species across 6,000 mammals evolved menopause, if the grandmother effect is as powerful as the evidence indicates, has a specific answer. The specificity matters because it tells us not just why menopause exists, but what exact conditions were required to produce it. Identifying those conditions reveals why the adaptation is so rare, and why its rarity is not a puzzle to be explained away, but a precise signal about what kind of place in the biological and social world is required before evolution can arrive at this particular solution. The answer consists of 3 necessary conditions, each well supported in the comparative biology literature, each independently constraining which species can potentially benefit enough from grandmothering to overcome the reproductive cost of stopping. And almost no species meets all 3 simultaneously. Humans and 5-toothed whale species, through completely independent evolutionary paths, apparently do. The first necessary condition is stable, multi-generational social group membership with high and persistent genetic relatedness between co-residing generations. This condition is the entry requirement for the inclusive fitness accounting that makes grandmothering evolutionarily worthwhile. Grandmothering increases a post-reproductive female's inclusive fitness only if the individuals she is helping are closely related to her, carrying significant fractions of her genes, and if they are reliably present in proximity to her on a daily basis over extended periods. In species with strongly dispersal dominant mating systems, where offspring leave their natal group at sexual maturity and spend their adult lives without regular contact with their mother or maternal relatives, a post-reproductive female simply has no closely related grandchildren nearby to help. She could invest enormous energy in the offspring of strangers, but inclusive fitness does not benefit from helping non-relatives. The fitness gains from grandmothering require that the grandchildren be present. Killer whales of the resident ecotype meet this condition completely. All individuals, male and female, remain in their natal group for their entire lives. So a killer whale grandmother is surrounded by her adult children and their offspring every day of her post-reproductive life. Human hunter-gatherer bands meet it in varying degrees depending on postmarital residence patterns, but the multi-generational household and residential cluster structures that characterize the majority of traditional human societies consistently place grandmothers in daily proximity with grandchildren. Most other mammalian species do not meet this condition. Some chimpanzees have female-biased dispersal, meaning daughters typically leave their natal community at sexual maturity. The grandmother-grandchild proximity that grandmothering requires does not reliably exist in chimpanzee social structure. Elephants maintain multi-generational family groups, and there is some evidence for a weak grandmother effect in elephant populations, but the conditions under which the effect operates are less extreme than in whales or humans, and the effect is correspondingly weaker. The second necessary condition is an ecological environment complex and variable enough that accumulated multi-decade experience of specific resource locations provides survival advantages large enough to be worth the reproductive cost of the individual maintaining that knowledge. This condition is the mechanism requirement for grandmothering to produce inclusive fitness gains, rather than just proximity. A post-reproductive female who is present near her grandchildren, but has no knowledge advantage over younger group members, provides caloric support if she can forage effectively. But she does not provide the specific, irreplaceable ecological intelligence that makes the grandmother effect most powerful. The grandmother effect is largest in killer whales and in human populations during periods of resource scarcity. That when the food environment has shifted in ways that require multi-decade experience to navigate successfully. During abundant years, when salmon are everywhere or shellfish beds are universally productive, the grandmother's specific locational knowledge provides marginal advantage. During lean years, when the food has gone elsewhere and only someone who has observed the food going elsewhere before, in previous lean years across multiple decades, and knows where it went. The grandmother's knowledge is irreplaceable. Species that live in simple, abundant, or highly predictable food environments do not generate large grandmother knowledge effects, because the knowledge that decades of experience provides is not much more valuable than the knowledge that a few years of observation provides. The environments in which killer whales and prehistoric coastal human women lived are neither simple nor predictable. They are variable, heterogeneous, so knowledge-intensive in exactly the way that makes a 50-year database more valuable than a 10-year database. The third necessary condition is the most demanding of the three, and it identifies the key variable that explains why only 6 species and not 60 meet the combined requirements. For stopping reproduction to be genuinely more adaptive than continuing it, for natural selection to actually favor the genes that produce cessation over those that produce continued reproduction, the post-reproductive female must be in a demographic situation where the inclusive fitness gains from helping her grandchildren exceed the direct fitness gain of producing one more offspring. This requires 2 things to be simultaneously true. Juvenile dependency must be high enough and long enough that the grandmother's help during the dependency period makes a meaningful difference in survival probability, and the grandmother must have enough living close relatives nearby that the total inclusive fitness benefit of helping them is large. Both requirements favor species with long juvenile dependency periods, low juvenile mortality with grandmother help compared to without, and high co-residence of multiple generations. Human infants are born with brains at approximately 25% of adult volume, the least neurologically developed neonates among all primates, requiring the most extended period of intensive caloric support before independence. Hodza children cannot meet their own caloric needs through foraging until approximately age 15. 15 years of nutritional dependency, and across which every increment of additional food provision from a grandmother translates directly into growth and survival probability. The marginal value of one more infant, competing with existing grandchildren for the grandmother's resources, and her attention is lower than the marginal value of the sustained, multi-year support she provides to grandchildren already in her care. The arithmetic favors stopping. A mathematical model of this condition, developed and published by Pia Smaldino and colleagues building on the Hawkes grandmother hypothesis framework, demonstrated through simulation that grandmothering behavior alone, without any additional mechanism, could drive the evolution of extended post-reproductive longevity from a chimpanzee-like life history baseline to a human-like one within approximately 37,000 simulated years. And the simulation required only that: a post-reproductive female provision grandchildren from foraging surplus, that this provisioning measurably increased grandchild survival, and that the demographic structure placed grandmothers in sufficient proximity to grandchildren for the provisioning to occur. Under these conditions, natural selection extended post-reproductive lifespan from approximately 25 years to approximately 49 years automatically, and because the genes that extended post-reproductive life were being spread forward by the same inclusive fitness accounting that the grandmother effect generates. The scientific debate that remains open within this framework concerns whether these three conditions are jointly sufficient to predict menopause in any mammalian species that meets all three, or whether the prediction requires additional specifications. Just some researchers working on the theoretical models argue that the conditions are necessary, but not jointly sufficient, that a fourth condition involving the specific ratio of costs and benefits under realistic demographic assumptions is required to push the fitness calculation past the threshold at which stopping is actually selected for, rather than merely neutral. So others argue that the three conditions are sufficient in principle, but that very few species meet all three simultaneously, with enough margin to generate selection, which is why the outcome is so rare. The 2024 Nature analysis of 32 toothed whale species supports the three-condition framework by showing that the 5 menopausal species share all three conditions: stable multi-generational groups, complex variable food environments, true and long juvenile dependency periods, while the 27 non-menopausal species consistently lack one or more of the conditions. The framework predicts the empirical pattern across the 32 species with enough accuracy to constitute evidence rather than merely hypothesis. 6 species across 6,000. The rarity of menopause across mammalian life is not a puzzle suggesting that the adaptation is fragile or improbable in principle. And it is a precise biological signal about how demanding the conditions for its evolution are, and about how rarely those conditions co-occur in the same species in the same place at the same time, with enough consistent intensity to drive the necessary selection. When they do co-occur, when a species lives in stable multi-generational groups, in a complex variable food environment where accumulated knowledge matters enormously during scarcity, with juveniles dependent long enough that grandmother provisioning makes a survival difference, evolution finds menopause every time, in killer whales and in humans, separated by 90 million years of independent evolution, in different oceans and on different land masses, in completely different ecological contexts. Selection arrived at the same answer. The answer is a post-reproductive female who knows what the group needs, who knows where it is, who has the time and energy to provide it because she has stopped investing in new offspring, and whose contribution to the group's survival in exactly the conditions where survival is hardest is measurably, statistically, and profoundly significant. The question stated at the beginning of this documentary was this: what evolutionary force was powerful enough to eliminate reproductive capacity in the middle of a woman's life, to override the foundational logic of natural selection, which selects with extraordinary consistency for continued reproduction? And what does the answer reveal about the conditions under which the human species narrowly survived its most severe encounters with extinction? Ideas that question has been held through 10 chapters of evidence. The answer is now complete. Approximately 150,000 years ago, the human species was compressed through a series of catastrophic environmental events. Mega droughts, severe enough to reduce East African lake volumes by 95%, vegetation collapse across the continental interior, and supervolcanic forcing that disrupted food chains already under severe chronic stress, into small coastal refugia along the southern and eastern shores of Africa. Within those refugia, bands of 25 to 50 individuals survived by exploiting year-round marine protein resources on coastlines that the mega-droughts inland reach could not destroy. They survived because they knew exactly where the calories were and exactly how to extract them efficiently. The knowledge of where the calories were was not evenly distributed within those bands. It was concentrated in the oldest women, the women who had been observing the same stretch of coastline for 3 and 4 decades, who remembered where the shellfish beds had shifted, the last time conditions changed, who knew the seasonal and annual patterns of resource availability that younger women had not yet lived through, enough cycles to have learned. The effective breeding population that survived the worst of these events may have been as small as 5,000 to 10,000 individuals. The entire genetic future of 8 billion human beings passed through that filter. The genes underlying the capacity to live productively for decades after last reproduction, the genes that encode what we call menopause, were selected in those groups during those crises, because the women who expressed those genes were the ones whose groups survived, and survival of the group was the test that selection administered across every generation of that period. The 290 genetic variants now measurable in the modern human genome, distributed across DNA repair pathways and reproductive hormone response systems, are the record of that selection. They are ancient. They are precisely tuned. They are present in every woman alive today whose maternal ancestors passed through those bottlenecks, which is, without exception, on every woman alive today. You carry this history in your biology with a specificity that should be stated plainly. The mitochondria in every cell of your body, those organelles generating the chemical energy that powers every thought you have had today, every step you have taken, every moment of attention and feeling and decision, trace their cellular lineage through an unbroken maternal chain to women who were foraging those sea cliffs when the interior of Africa was burning dry, and their choices, the directions they walked, the children of their children they fed, the knowledge they held and transmitted, the groups they kept alive through conditions that ended every other group around them, selected for the biological architecture that now determines the hormonal contours of your life. The timing of your menopause is a genetic program written under conditions of maximum evolutionary pressure by the most consequential selection event in the history of modern human biology. It is not a malfunction. It is not the body's apology for outliving its reproductive purpose. It is the inherited signature of women who were more valuable to the survival of their lineage after reproduction ended than during it. The women who accomplished this left no names in any record that survived. There are no monuments. There are no histories. There is only the mitochondrial phylogeny, the family tree written in the DNA of every person alive, tracing every maternal line back to the same narrow geography, the same narrow window of time, the same catastrophic filter, and the biology of the post-reproductive female body, shaped by selection over tens of thousands of years into a specific configuration for a specific function, not diminished, redirected, not past its purpose, so converted to a different and in those conditions more critical one: knowledge, navigation, social memory. Caloric surplus directed toward dependent grandchildren The accumulated intelligence of a full lifetime, freed from the metabolic and temporal demands of reproduction, made available to the group that needed it most during the years when the world was providing the least. There is a female killer whale alive in the Pacific Ocean right now who stopped bearing calves several decades ago and has been leading her family group toward salmon through 40-odd years of changing ocean conditions. During the lean years, when the salmon runs shift and the younger members of her pod cannot find the food, she knows where it went. She has seen this kind of lean year before, more than once, and she has learned from each one. She is doing what she evolved to do. She is doing what the oldest women on the African coastline were doing 150,000 years ago, in a different ocean, on the other side of the world, at the founding moment of the biology every human being now carries. The solution that evolution found independently, twice, in two completely different lineages across 90 million years of separation, is the same solution: an older female who has stopped reproducing, who knows things that took a lifetime to know, who is still here, still present, still providing what the group needs. That solution is not an accident of survival. It is the most tested, most independently validated adaptation in the history of our kind. The present moment, this room, this body, this night, is the direct consequence of the fact that those women did not stop contributing when they stopped reproducing. They became something different, and the difference was what the species required. Like whatever the biology of your own life holds, whatever transition you are in, or approaching, or carrying the memory of, you carry it in a body shaped by the most extraordinary, ordinary act in the history of human survival. An older woman walking toward the sea in the gray light before dawn, going where she knows to go.